New research shows that coronavirus has an entire second way, a second route of attack for getting into cells. Hello, everyone. I want to report on here today, but this is probably the most important, so we'll start here. I'm titling this, well, crap. It turns out that the coronavirus could attack the immune system like HIV by targeting protective cells. These would be the T cells. And researchers in China and the U.S. find that the virus that causes COVID-19 can destroy T cells that are supposed to protect the body from harmful invaders. One doctor said concern is growing in medical circles that the effect could be similar to HIV. So here's what that article from South China Morning Post, let's read through that, and then I'm going to show you the science behind it, the study that this is referring to, because I found the study. Let's see. The surprise discovery made by a team of researchers from Shanghai and New York coincided with frontline doctors' observations that COVID-19 could attack the human immune system and cause damage similar to that found in HIV patients. So what they did was they basically put this living virus, SARS-CoV-2, the honey badger virus, on laboratory-grown T lymphocyte cell lines. These are cell lines that they can grow, and you can grow and keep growing them. You don't have to harvest them from fresh patients all the time. They're lab cells used for all sorts of studies. T lymphocytes, also known as T cells, play a central role in identifying and eliminating alien invaders in the body. That's why if you attack the T cells, it's a very bad outcome usually, and that's what HIV showed. They do this by capturing a cell infected by a virus, bore a hole in its membrane. This is how the T cells protect things. I'll just be clear about that. And then they inject toxic chemicals into the cell so they kill the cell. And these chemicals kill both the virus and the infected cell and tear them to pieces. To the surprise of the scientists, the T cell became a prey to the coronavirus in their experiment. They found a unique structure in the virus's spike protein that appeared to have triggered the fusion of a viral envelope and cell membrane when they came into contact. So normally, what we're familiar with is that this spike protein binds with the ACE2 receptor, extremely high affinity. It looks like it might be binding to a second receptor. That's where I put the crap up there. All right, let's take a look at these cells. So notice here again on the ACE2 cell, it's binding in here with an infection ability that comes in at call it 6,000 just to be even, take even numbers. The infection of the MT2 cell line, which is a T cell line, for SARS-CoV-2, it had an infection ability of about 2,000, whereas SARS itself, 0, Mach 0, right? And here on this other one, the A3.01 cell line, a binding ability of about 1,200 or so, I would guess. SARS, pretty much 0, Mach 0. Okay. God, nuts. So that means that SARS, COVID-19 is coming in to T cells directly, at least in the lab. So we're going to see that this all gets followed up. By the way, this paper was in Nature, so I'm tending to trust this one quite a bit. Here's the punchline. This is from that same Nature paper here. They say, in other words, these results tell us that T lymphocytes may be more permissive to SARS-CoV-2 infection and less permissive for SARS-CoV infection. Okay. Therefore, it's plausible that the S protein of SARS-CoV-2 might mediate potent infectivity even on cells expressing low ACE2 receptors, which would in turn explain why the transmission rate of SARS-CoV-2 is so high. Why? Because it can bind to not just one but two separate receptors. It is also possible that the other receptors mediate entry of SARS-CoV-2 into T cells such as CD147, which is present on the surface T lymphocytes. So when we said ACE2, that's a receptor. CD147 is another one. So these are just some numbers and letters we're going to have to get used to, all right? Which was recently reported to be a novel invasive route for SARS-CoV-2, CD147. Really, so I had to chase that down. I clicked on number 9, got over here to this paper, which says SARS-CoV-2 invades host cells via a novel route, the CD147 spike protein. Currently, COVID-19 caused by severe acute respiratory syndrome, coronavirus 2, SARS-CoV-2, has been widely spread around the world. Nevertheless, so far there exists no specific antiviral drugs for treatment of the disease, which poses great challenge to control and contain the virus. Here we reported a research finding that SARS-CoV-2 invaded host cells via a novel route of CD147 to spike protein. Those two things were binding. SP-bound CD147, a receptor on the host cells, thereby mediating the viral invasion. Oh, Well, where else do we find this CD147? Hey, that's a good question. What's interesting, it's also known as Basingen. It's a protein that in humans is encoded by the BSG gene. This protein is a determinant for the OK blood group system. So it's here's where it gets real fascinating. There are 3 known antigens in the OK system, the most common being OKA, called OK1, OK2, OK3. Basingen has been shown to be an essential receptor on red blood cells for the human malarial parasite Plasmodium falciparum. So now we're starting to see a possible mechanism here for how this virus might even get into the red blood cells. And I know that there's been a lot of dinging of the so-called heme thing in that theory. Maybe that's the case. Maybe it's not. But at least now we have a way that we're starting to see some overlap here. And one of the more important overlaps, I think, that we're going to see is that the way that chloroquine and hydroxychloroquine work to prevent the growth of Plasmodium falciparum, the malaria parasite, is that the malaria parasite has a lysosome inside of itself. It's a membrane bucket, right? It's a membrane-bound vesicle that inside of which is where it puts the hemoglobin that it's eating out of the red blood cell to break it down. The lysosome, lyse, to break. That's what it means, to lyse. So it's breaking down the red blood cell hemoglobin in this lysosome. The chloroquine gets in there and it disrupts the pH of that thing. And so all of a sudden that lysosome doesn't work anymore. What's interesting is the lysosome is the same thing that when the virus binds onto either the ACE2 or the CD147 membrane receptor, that gets internalized, gets brought into a lysosome too, and then that lysosome starts breaking things down, and that actually liberates the virus into the cell, so the cell damage happens. So maybe it's possible that the chloroquine is doing something to lysosomes directly, and that starts to explain its connection here more than the blood thing. So we're going to continue to hack through this and figure this out as we go through it. Bottom line, though, is you really don't want this. More and more evidence is coming out that for the sickest patients, infection with a new coronavirus is proving to be a full body assault, causing damage well beyond the lungs. And even after patients who have become severely ill have recovered and cleared the virus, physicians have begun seeing evidence of really lingering effects, maybe permanent effects. In those who had survived mild and severe disease alike, researchers found that many biological measures had failed to return to normal. Chief among the worrisome test results were readings that suggested these apparently recovered patients continued to have impaired liver function. And that was the case even after 2 tests for the virus had come back negative and the patients were cleared to be discharged, so liver impacts. At the same time, as cardiologists are contending with the immediate effects of COVID-19 on the heart, they're asking how much of the damage could be long-lasting. In an early study of COVID-19 patients in China, heart failure was seen in nearly 12% of those who survived, including in some who had shown no signs of respiratory distress. Very troubling, and we're going to have to keep an eye out for that. Remember, as we push forward on this, the economic impacts are really just starting. They're going to be quite profound. Conclusions for today, these draconian governmental responses are quite concerning. Civil liberties are very easy to take away, it seems, and very hard to get back. That's been our experience through the last 20 or 30 years. The economic stimulus, though, by the United States, just a case study and too little, too late at this point in time. There's really no way to turn that ship around, I don't believe. So lots of economic disruption coming. COVID-19, God, this is really disappointing. Attacks both the ACE2 and, whoops, that was supposed to say CD147. I think that's all caps. That's secret all caps for 147 there. Receptor. Are there any others? We don't know yet, but if it's attacking the white blood cells, the T cells in particular directly, that's a really nasty finding. Explains a lot. And the damage here can be very long-lasting, maybe even permanent. So that's also a very disappointing finding. And remember, based on this, so with that, I've got to run today. I've got a TV interview coming up shortly on Fox After the Bell. So let me run to that. I'll be back with you tomorrow. I got some really exciting findings from some autopsies that came out. I know that sounds an awkward sentence, right? Exciting findings, but it's giving us some real clues as to what's actually happening here. This is a real honey badger of a virus and does awful things, and we're learning more and more by the day. Thank you for listening. Talk to you tomorrow.
Hi folks, Adam Taggart here. Chris Martenson and I are the co-founders of Peak Prosperity. If you want to get alerted whenever we release a new video from Chris, just click the red subscribe button right beneath the YouTube video player. Once you've done that, a little bell icon will appear right next to it. Click on that bell. It looks like this.